off. As I said before, she was not able to be, Erica was not able to be with us today. So if you have any question, you can directly go on Discord to ask her. And so now we will pass to our next talk. Who should arrive in the breakout room very soon. Hi, William, can you hear me?
I can.
So we will have a presentation from William Botta, who will talk about panic in the dark, factor underlying the spread of collective fear in the zebrafish Danio area. So William, I guess you know the saying, with great power come great responsibility. And tonight your power is that you are the last talk and your responsibility is to actually close this conference for this year. So good luck with that, and people, I would say, enjoy as much as possible this last talk, as you will have to wait one more year to have more of all this science. So the floor is yours, William. You can just share your screen.
Perfect. Everybody see it? All right. Hi, my name is William Botta. I am
William. Sorry, William. We have the camera that are from Zoom that are on your screen right now. I don't know why, but
like it's doing what?
Yeah, it's like if Zoom was before your presentation. Basically, we can see your control, your Zoom control, as well as your camera.
Oh, okay, yeah, like that, right?
Yeah.
Cool. Yeah, I don't know why it does that. Yeah, so my name is William Botta. I'm a second-year PhD student in the biology program of New Jersey Institute of Technology, and I will be telling you about factors underlying the spread of collective fear in the zebrafish Danio rareo. So, before we get into that, there we go. Let me define what panic is. Panic is a sudden, overpowering fear response resulting in a flight response. Basically, an organism senses a threat and becomes overwhelmed such that it can only respond with running away. Mass panic is going to be the spread of that panic response within a group or population. Basically, one organism sees a threat in its group, and then it becomes overwhelmed and flees, and the rest of them sense that, and that's how it spreads throughout the group. This can be useful because it allows an entire group to be made aware of a threat and act accordingly all together. Here we have a GIF of a school of fish where they see a shark coming through and they all get out of the way so that they don't get eaten. Down at the bottom, we have a herd of buffalo that they also sense some sort of threat, and it allows them to all move together so that nobody gets left behind. However, mass panic isn't all good things. Unfortunately, unchecked panic can lead to poor collective decisions. If one person at a concert rushes the stage, other people might see that and think, oh, now's my chance to rush the stage as well, and everybody makes that decision together. If this feedback loop breaks, panic might not stop. It might cause tramplings as people try to escape a theater, even though a perceived emergency situation isn't as bad as it might have actually been. So because of all of these potential negative effects of a panic, it's important to understand what causes them and why they might occur. So my big question is, what are the factors that control the cause and propagation of mass panic? In general, there are environmental factors and neurobiological factors that might cause this spread. A lot of work has been done in looking at how environmental factors play a role in mass panic mitigation. Tight passageways and obstructions in front of exits actually reduce panic and allow for a more controlled exit of a stadium or other large arena. However, less work has been done in the neurobiological factors of mass panic. Things such as how might sight and touch and other senses affect the spread of panic? What about mental state or anxiety level? Are individuals who are more anxious more likely to panic? And these lead us to ask questions about how we might test these factors. Finding a good model organism to test can be difficult. They need to actually exhibit a panic response, but they also can't be too large because observing all of the individuals in large organisms can be difficult. Inducing panic in a large organism might require a lot of space. And testing in the most important organism to us for this, humans, has a lot of ethical implications. We can't exactly put 200 people into a room and say, everybody panic, and here's some drugs to affect that panic. It would be a madhouse. So, in searching for a good organism, we've settled upon the larval zebrafish, which previous studies have shown to exhibit a mass panic-like behavior, such as what is going on the screen right now. They've been found to exhibit this weird panic-like behavior in sufficiently high densities where they all swim around together around the outside of the arena, and then they all stop, and then they start back up again. They're a great organism to test this stuff with because they are a model organism. We can play with their genetics to alter their behaviors and also light up neurons in their brains so that we can see how they're firing. They're easy to grow in large numbers, and they're transparent, so we can actually see those brain responses live while they're exhibiting their behaviors. They're a good alternative to humans because we can apply drugs to them. Here's another example of a panic. I love these videos because they just kind of go. And so they're a good alternative. We can apply drugs to them to alter their mental states in a non-invasive way. And they're just very easy to work with. They're very nice and small, so we can take very quick videos of them easily. So, in our first step is identifying conditions in which zebrafish panic occurs. Now, previous studies have found that density of population is a major driver of these panics. We tend to see that these mass panics don't really occur until we hit a density of at least 5 fish per square centimeter of area. That's 130 fish in a petri dish of 5.5 centimeters. Now, when we look at a zebrafish, we see two big things that the two biggest things that jump out of a larva here are going to be its long tail that it uses for moving around and catching food, and then its eyes, which are super large and very clearly important. So this vision has to be one of the most important senses because of that. So our questions are, is there a way to make these panic events more consistent? Is it going to be due to some sort of sense with their vision? And how do the effects of these environments contribute to consistent panic? So in order to test these, we took 5 and 6 day old larval zebrafish, and we recorded them at 200 frames per second in arenas of varying size, keeping the density of fish consistent at 8.5 fish per centimeter squared. So we might take a video of 200 fish in a 5.5 centimeter diameter container, and then in a container a quarter of that diameter only have 50 fish, or not a quarter of that diameter, a quarter of the area. These experiments were conducted during periods of constant light or constant darkness to see how much vision and being able to see would play into these panic events. And then some of their arenas had their centers filled in to prevent fish from staying there. As you see, during these panic events, all of the fish are circling around the outside, but then there's still some in the middle that are resting. So we wanted to see how something like a central obstruction might mess with that behavior. So we took these videos and then we analyzed them for motion measuring using optical flow, which compares groups of pixels between frames to estimate if and how much motion may have occurred without the need to actually track each individual fish. These were then filtered for noise using fast Fourier transformation. We then proceeded to test for panic propagation using connected components. If you want to give a minor flashing light warning, there's just going to be white dots appearing randomly in this video. But essentially what it does is we use a thresholding algorithm to calculate areas of high motion, which are represented by these white dots. And then we can track from frame to frame whether white dots in one frame are close to or far from the white dots in another frame to basically estimate whether or not that motion in that frame is being caused by the motion from the previous frame in each video. And we then used all of this information to compare the amount of motion in light versus dark conditions amongst the different arena conditions. So our preliminary results seem to show that fish are much more likely to experience these mass panic events in the dark than in the light. In my graphs here, we have time represented on the x-axis and amounts calculated on the y-axis. And the most striking thing here is just how much more motion there is in the dark compared to the light. Each one of those repeating peaks in our dark experiments here corresponds to a panic event that occurs in our videos. And we can see that in the dark, in all of our conditions, we see these panic events, whereas in the light, they tend not to panic as often or as intensely. So this would suggest that vision appears to be a major modulator of this behavior. When looking at the central obstruction analysis, we found that central obstructions do not appear to have much effect on the panic. Here we have the different populations in their respective arena sizes on the x-axis, and then the total motion calculated in each video on the y-axis. And we can see here that regardless of whether the ring was present, as in the blue boxes, or absent, the central obstruction was present in the blue videos versus absent in the red videos, there does not seem to be much difference between those videos. However, in the light versus darkness experiments, we see that amongst our different populations, in general, the dark videos had much higher total motion versus the light. This is especially pronounced in our largest arenas at the 200 level, where on average, there was almost double the amount of motion in the dark videos versus the light. However, outside of the 200, the largest arenas, our arena size did not seem to play much of a role in determining how much panic would occur. So our major conclusions are that vision seems to be an important factor in the propagation of mass panic. Their ability to see their conspecifics might be modulating the escape response that they produce when they perceive a threat, causing them to not swim into each other, causing them to induce their own panic events. Whereas in the dark, being unable to see where they're going might play an important role in their lack of modulation there. We also see that panics can be induced in arenas of varying size, so density is the limiting factor there. There also seems to be a refractory period after each panic. So we see these mass panic events as these peaks, and then afterwards, all of the fish seem to stop moving for a time or move very little before jumping right back into a new one. So they are resting. And then because of this refractory period, there seems to be some periodicity. We have peaks that are very almost equidistantly apart from each other. So there might be something there in terms of how panic can repeat and propagate over and over again. So with all of these conclusions that we've drawn, there are some obvious next step questions that I will be hopefully testing in the future. We want to look at other senses and how they might play a role in this panic. Fish have a lateral line organ, which allows them to sense changes in water pressure as they as it flows around them, and this can give them a sense of things that are moving nearby and currents. So we want to test ablating that lateral line organ to see if that is an important mitigating factor in these panic events. We also want to test that periodicity that we observed in these videos, where we can disrupt that cycle. We have created a device in our lab that can basically produce these sounds that have been shown to cause zebrafish to elicit that fast escape response. And if we were to use that on the Petri dish containing all of the fish, they would all produce that escape response at once. So we could test disrupting that cycle by exposing them to that sound during a panic or during a refractory period to see how that would change their behavior. We'd also like to look at differently shaped arenas to see how limiting their ability to see conspecifics via like environmental conditions, such as a random forest of pillars in the Petri dish, might change their panic patterns. And then we want to look at mental state, inducing anxiety with anxiogenics and relaxed states using anxiolytics to see if the introduction of calm individuals makes panic more or less likely to, or anxious individuals more or less likely to produce these panic effects. I'd like to take a second to thank a bunch of people, including the Animal Behavior Society, for providing a grant that I am using to fund this research, as well as my PI in the Swarm Lab, Simone Garnier, as well as the various other members of the lab for their assistance with this, and then my PI in the Severi Lab, Kristen Severi, and everybody there for all of their help in the research and the preparation of this presentation. Thank you guys so much. And at this point, I would like to take any questions.
Thanks a lot, William. So we already actually have some question in the chat. So we have Noah Fu who say, why will narrow escape routes reduce panic? Is it because it forces the fleeing individual to slow down? Wouldn't a narrow limited escape increase panic due to limited escape route?
Right. So I'm going to say part of it is limiting the amount of motion they can actually do, as well as limiting the vision, like the ability to see what other conspecifics are doing. If you can't see other people panicking, you are less likely to panic yourself. And then also in terms of, let me go back to like close to the beginning, this one here, producing an obstruction like this opens up multiple path points. So even though you're all going through the same actual exit, you feel like you're making more progress. And so that reduces your panic state.
Okay, thank you for the answer. We have some reaction comments like, thank you for compelling talk. We have from Diane K. And we have Santiago Meneses that clap his hands for your talk. I have myself one question in two parts. I mean, depending actually on your first answer. I was not able to determine really how long was the refractory period in average.
Oh, let me go back to the graph. Here we go. It would depend on the on each video. It was a little different. Let me go back to this one specifically. So if each of these, so this is the frame number in the video. So at 200 frames per second, this one's occurring at roughly 2000, maybe 3500 frames. If I were to do that. That's going to be, doesn't seem right. Maybe I'm bad at math. That's what that means.
So like you say it's 200 per second.
Yeah. So each frame is 1/200th of a second.
So like, I mean, it's not that long in the end. I thought that it was a way longer period than this, because like my hypothesis that like maybe because we know that cortisol in water takes about 2 hours, 3 hours before to dissipate. So that maybe your refractory period was observed because of this cortisol in water.
I'm sorry, could you repeat that?
Yeah, I said because like I was asking this question about the time of a refractory period, because like if it was a kind of a long refractory period, my hypothesis that maybe it was the cortisol that stained the water that could actually trigger this refractory period. Did you try to test the level of cortisol in water during panic or these kind of things?
I have not, but that is definitely something that we can take a look at.
Yeah. And so we received some other questions. One from David McCluskey. How applicable are the conditions for panic, poor light density, etc., to other organisms, bird, bigger, fish, human?
I would say that, like, especially with light, that's going to be very applicable to any organism that lives in a group that uses vision as one of its primary sensory components. Ability to see your conspecifics and act accordingly is going to play a major role in the behavior that you induce. And then group density, that's definitely going to affect how easily you can like sense your other conspecifics in other ways. If the density is low and you feel a threat, you perceive a threat, and the density is low, it's going to be difficult for that information to get transmitted through the group very quickly versus in a higher density environment. If you see a threat, you immediately can tell someone who's standing right next to you. So,
yeah, thanks.
Yeah.
So like we have two last questions. I think it will be the two last one for this talk from the same person, A.B. George. So first he says, so great talk. Nice tie-in with Einon's talk on the first day on how the shape of arena affect movement patterns. Oh yeah, so I think what he's saying is that like there is a correlation between your talk and the talk of Einon earlier yesterday about the fact that the shape of arena were actually affecting the movement. But he asked a question: can you explain the choice of density a little bit more?
Sure. So this was the original paper, Zaki et al. 2021. They were just, it was kind of a shot in the dark. We realized that we had never put zebrafish in high densities in the lab before. So they tested zebrafish at a variety of different densities from 5 all the way up to 130, just to kind of see what they would do. and they saw this unique circling behavior that we'd never observed before. And so they put out a paper on it because it was cool and exciting. And when we noticed this, we realized that it could be a major model for panic behavior in general.
Okay, thanks for the answer. And thanks, William, for your talk. That was really interesting. If some of you have more questions for William, like you can go on his Discord channel. And so like you will be able to find the talk anyway during 2 weeks. So yeah, thank you, William.
Thank you.